Hello students, in our previous chapter of 12th standard biology, that is Principles of Inheritance and Variation, we have studied the experiments of Mendel, Morgan, Sutton and Boveri. Going by their observations, we know that characters are inherited in organisms from one generation to the next. But by their observations, we still do not know which is that molecule that plays the role of genetic material. That is, it was not known what is the biochemical nature of the genetic material. And that is how it became very much necessary to bring about the search for genetic material. So here we begin with a new topic in our next chapter, that is molecular basis of inheritance. In that we are going to study about how the search for genetic material began. The very first experiments in this direction were performed by Griffith. Griffith in the year 1928 performed his experiments on the bacterium Streptococcus pneumoniae. This bacteria he cultured in labs using culture plates. He found that the bacteria has two types of strains, that is S strain and R strain. S strain is the smooth strain, whereas the R strain is the rough strain. S strain is called as the smooth strain because when it forms colonies, the colonies appear smooth. That is, the bacterial colonies have cells which are surrounded by polysaccharide coats. Due to the presence of polysaccharide coats, the bacterial cells get a smooth appearance and thus their colonies appear smooth. Whereas the rough strain bacteria do not have the polysaccharide coat, due to which they do not have the smooth covering and so they appear rough. He performed his experiments using both the strains of the bacteria. The first step that he did was the S strain bacteria was taken and injected into living mice. When this strain was injected into mice, he found that the mice died because the mice got infected by pneumonia, which is caused by Streptococcus pneumoniae S strain. So he concluded from this step that the S strain can be called as the virulent strain. Virulent means the one which is capable of causing the infection. Later, in the next step, he this time took the R strain of bacteria. The R strain bacteria he injected into mice and he found that the mice were living. They did not die due to infection. So, the R strain bacteria are said to be avirulent. In the third step, what he did was, he took the S strain bacteria again, but this time he heat killed the bacteria. Means he heated the bacterial cells, due to which the cells died. So, the S strain heat killed bacteria were injected into mice. This time he found that the mice were alive. They did not get the infection because the S strain bacteria were heat killed. In the further step, he took the S strain heat killed bacteria along with rough strain, that is R strain living bacteria. Remember here the S strain is dead whereas the R strain is alive. He mixed both the strains and this time he injected the mixture of the two strains into mice. This time he found that the mice died due to infection. Means by observation of this result, he concluded that there was something in the S strain which transformed the R strain, due to which the R strain, which was earlier incapable of causing the infection, got transformed and now became capable of causing the infection. This particular material which was transferred from the S strain to the R strain was named as transforming principle. So the transforming principle is that material which was transferred from S strain to the R strain and brought about the transformation. What is transformation? Transformation means the genetic alteration of the cell due to uptake of a genetic material. So with this experiment, he could show that something was transferred from S strain to the R strain, which was capable of bringing about transformation. But that something, what was it actually in the biochemical form, that could not be told. So still, the biochemical nature of the genetic material was unknown even by this particular experiment. Further experiments were performed by three biologists, that is Avery, MacLeod and McCarty, through the years 1933 to 1944. They made use of the same kind of bacteria that was used by Griffith, that is Streptococcus pneumoniae. In this bacteria, they utilized the S strain. The S strain of the bacteria was taken and the bacterial cells were heat killed. From the heat killed S strain, they isolated three kinds of biomolecules, that is proteins, RNA and DNA. The protein molecules were mixed with the living R strain and the effect was observed. With the help of this observation, they concluded that the R strain could not be transformed where proteins were added into it. Thus, protein was not responsible for bringing about the transformation. Next, they took up the isolated RNA molecules and mixed it with the R strain. When they observed the results, they found that still no transformation took place. Means RNA was also not responsible for bringing about the transformation. Further, they took the isolated DNA molecules and mixed it with the living R strain. This time the result was transformation had occurred. From this, they could say that DNA was responsible for bringing about the transformation of R strain. They did not stop here. They went further, and what they did was, the protein molecules were treated with enzyme, that is proteases. Proteases are those enzymes which break down proteins. So, the treated proteins, that is the digested proteins, were now mixed with the living R strain. Still there was no effect observed. Further, the RNA molecules were treated with the enzymes, that is RNases. RNases are those enzymes which break down RNA. So this treated RNA molecules, that is digested RNA molecules, were mixed with the living R strain. Still there was no effect. Further, the DNA molecules, they were treated with the enzymes, that is DNases. DNases are those enzymes which break down the DNA molecules. So, the treated DNA molecules were mixed with the living R strain, and this time what they observed was no transformation took place. Remember, the earlier observation was DNA brought about the transformation, but the same DNA molecules, when they are broken down and then added to the R strain, they could not bring about the transformation. So with the help of this observation, again they could say that DNA was responsible for bringing about the transformation, and thus DNA could be called as the genetic material. But still, even after these observations, many of the biologists were not convinced. And now finally, the experiment that led to the proof that DNA is the genetic material. This experiment was performed by two biologists, that is Hershey and Chase, in the year 1952. In their experiments, they made use of bacteriophages. Bacteriophages are those organisms which are actually the viruses that infect the bacteria. These bacteriophages, when we consider their structure, they have two portions. That is the outer portion, which consists of protein coat, and the inner portion, which consists of genetic material. They used the bacteriophages in the labs by growing them in two different culture medium. In the first culture medium, they made use of radioactive sulfur. Now why do they make use of sulfur over here? Because viruses need sulfur in their protein coats. The protein coat shows the presence of sulfur, whereas the genetic material, that is the DNA, shows the presence of phosphorus. The point to remember over here is proteins have sulfur but not phosphorus, whereas DNA has phosphorus but not sulfur. So they grew these bacteriophages in two different culture media in the labs. In the first culture medium, they added radioactive sulfur, that is 35S sulfur. The new viruses that will grow in this particular medium, they will show presence of radioactive sulfur in their protein coats. Whereas the viruses that will grow in the second culture medium, they will show presence of radioactive phosphorus in the DNA. Why? Because in this culture medium, they have added radioactive phosphorus, that is 32P phosphorus. And remember, phosphorus is present in the DNA. It is not found in the protein coat. So when we study their experiment using the viruses of the first culture medium, this culture medium, as I told, they have added radioactive sulfur. So in the protein coat of the virus, we should have radioactive sulfur. This virus, that is the bacteriophage, which was grown in radioactive sulfur, was allowed to infect the bacterial cell. When this bacteriophage infects the bacterial cell, what it does is, it leaves the protein coat outside and inserts the genetic material on the inside of the bacterial cell. What is to be remembered? The protein coat is left outside and the genetic material goes inside the bacterial cell. Okay. When this infection was done, as we know that the protein coats will be left outside, so they took this mixture of the bacterial cell along with virus particles and agitated this mixture in a blender so that the viral protein coats could be separated from the infected bacterial cells. Further, they allowed this mixture to undergo centrifugation, that is a separation technique. When the centrifugation process was completed, two layers are obtained in the centrifuge tube. The upper layer is known as supernatant layer. It consists of the viral protein coats. The lower layer consists of the bacterial cells which are infected with the viral genetic material. Both the layers, that is the upper layer and the lower layer, were tested for radioactivity. When they tested the upper layer, that is the supernatant layer, what they found was radioactive sulfur, that is 35S sulfur, was present in the upper layer, that is the layer of viral protein coat. But when they tested the lower layer, they found that there was no radioactivity. Why? Because the lower layer has bacterial cell with the genetic material. It does not have viral protein coat. With the help of this particular experiment, they could make out that DNA is inserted, but the protein coat does not get inserted. It remains separate from the bacterial cell. Still, they carried on the experiment in the next part. In the next part, what they did was, they took the bacteriophages, that is the viruses, which were grown in a medium containing radioactive phosphorus. These bacteriophages, we need to remember, are grown in radioactive phosphorus, that is the phosphorus which is present in the DNA. So DNA should have radioactivity, whereas the outer protein coat will not have any radioactivity, because protein coat does not have phosphorus. It is the DNA which has phosphorus. These bacteriophages were allowed to infect the bacterial cell. We know how the bacteriophages infect the bacteria. They leave their protein coats outside and they insert the genetic material on the inside. So bacterial cells, when they are infected with virus, that is bacteriophage, they receive the viral genetic material, but the viral protein coats are left outside. Further, they put this mixture of the bacterial and the viral cells into a blender. It was agitated so that viral particles could be separated from the bacterial cells. Further, they allowed this mixture to undergo centrifugation. When centrifugation process was completed, again here they will get two layers. The upper layer, which is the supernatant layer, which will have the protein coat of virus. The lower layer, which is the layer of bacterial cells which are infected with viral genetic material. So, when these two layers were tested for radioactivity, it was found that the upper layer, that is the layer of protein coat, did not show any radioactivity. Whereas the lower layer, that is the layer of bacterial cells, along with viral genetic material, showed the presence of radioactivity. Thus, it showed the presence of radioactive phosphorus. Okay, with the help of this, we can say that the DNA of the virus, which was having radioactive phosphorus, was transferred into the bacterial cell at the time of infection. That is, the virus or the bacteriophage transfers DNA into the bacterial cell. So it is actually the DNA which gets transferred from the bacteriophage into the bacterial cell, and thus it was detected over here. This experiment was the ultimate proof that DNA is actually the genetic material. So by now, I hope that you have understood the search for genetic material involved which all experiments, and ultimately it was the Hershey and Chase experiment which proved that DNA is the genetic material. Thank you.